Well, good morning or good evening, whenever you're watching this. I wanted to give you a head start a little bit on the lipid section on Chapter 3, but before I do that, I want to look at the end of Section 2, because we've mentioned a little bit about these things called enzymes, and some of your experiments that you're using with carbohydrates are actually looking specifically at one type of an enzyme, that enzyme being named lactase, the one that actually breaks down the sugar lactose into its two components, which is glucose and galactose. So, but one of the things I just want to bring up here is that in order for a lot of these reactions to happen, in order for hydrolysis reactions to happen and dehydration reactions to happen, so breaking down or building up new molecules, we need to use enzymes because they greatly facilitate the speed at which this happens. So a couple things here. Dehydration reactions form new bonds. So the forming of new bonds is going to require energy, where hydrolysis is going to break bonds, and that's going to release energy. So just a couple things to note. Here's some examples of some specific enzymes for different macromolecule classes. So we mentioned lactase, sucrase, amylase, maltase. So a lot of the enzyme names are very specific to the substrate or the thing that they actually break down or work on. So we're about to get into lipids, and lipids have a group of different enzymes called lipases, because lipid is a general name for fats, and lipases are the enzymes which will be involved with breaking them down or building them back up. And then finally we'll look at proteins and proteases of different types. Before I move into lipids though, just a couple things on polysaccharides. So we mentioned starch, and there's a couple different versions of starch that we can see. And the way these starch individual monomers join is by these glycosidic bonds, just like every other monomer forming a polymer. But here I just wanted to point out to you this idea of what we're calling alpha or A, 1 to 4 glycosidic bonds, or alpha 1 to 6 glycosidic bonds. So those numbers, the 1 and the 4 and the 1 and the 6, are indicating the carbon, the actual numbered carbon that is doing the bonding with the adjacent molecule. So the 1 to 4 means that it is a bond between a carbon number 1 on one molecule and number 4 on the other. So here in this example here, we could say that this glucose right here, this represents the number 1 carbon, and this represents here on the adjacent molecule the number 4 carbon. And it's an alpha bond because of the fact that it's going down. So when we see the bond going like this, that's an alpha. Where if it was going up, that would be called a beta. So it's just recognizing the direction that bond is, whether it's up or down. So an amylose, which is a starch, it's unbranched, and you're going to see that 1 to 4 glycosidic bond, the alpha type, throughout. So that's the top picture, where amylopectin is below, and that is a branched structure. And notice what happens here. On that one, the individual chains are 1 to 4 glycosidic bonds, but because there's a branch there too, then we're seeing on this one right here, this 1 to 6 glycosidic bond. So that's and they're both alpha because they're going on this primary carbon is going down. Cellulose is also a type of polysaccharide, and if you notice what's going on here, you can kind of see that each of these individual monomer units becomes actually flipped. So we have, here's that, look for this to really show that. So that this part right here, the CH2OH down here, is on the bottom, now it's on the top, now it's on the bottom, now it's on the top. So these glucose monomers it's unbranched, so it's linear, but there are beta 1 to 4 glycosidic linkages. They're beta ones. Now they're beta, you might say, well, wait a minute, how do I know that? Because it's the direction of the molecule itself, which is flipped, but these bonds that you see here are actually more like this, except for we have that flipped part on that one next door. So this is a beta. Now, what's interesting about cellulose is that it is a polysaccharide, so it's like a starch. But for us, we cannot break it down because we don't have the enzyme to break down cellulose. Where we do have enzymes to break down the starch molecule. And so that is something we can take advantage of and get energy from by that enzyme helping with hydrolysis of those big long chains. All right, chitin I just mentioned briefly. It's no more to be said really than that. So I need to keep moving here. So lipids are fats. I mean, it's AKA, that's what we typically think of, although lipids are much more diverse than just the dietary fat that we think of. And they're nonpolar, therefore they're hydrophobic. And that's really an important thing because we're going to see why as we go forward. Lipids are great for energy. As a matter of fact, if you eat per gram, like butter, for example, or some other lipid, you're going to get over twice as much energy than you would for an equal gram of a carbohydrate or even a protein. So it's a really high energy source. It's used for insulation, in other words, for the fat that keeps our organs warmer and our bodies warmer. It's a building block for hormones, and it's a really important component of cellular membranes, as we're going to see. So here's the types of lipids that we can look at and will look at: fats, oils, waxes, phospholipids, and steroids. Fats and oils are the first one, and they have two main types of units. One is called glycerol, and one are called fatty acids. And when they come together, most of the things that we see in this category are called triacylglycerol. And the reason they are is because they use actually 3 of these chains of fatty acid for every 1 glycerol molecule. So I'll show you that in class. And we have this special attachment. Now we have a new bond that forms in lipids called an ester. So an ester linkage is what we're going to be looking at with fats and with this particular type that we're looking at here. So instead of a glycosidic linkage, we're looking at an ester linkage. And when that happens, because there are these 3 chains of fatty acids that are joining, we're going to lose 3 molecules of water. Here's the good picture, though, to kind of show that. So glycerol at the top, notice it has that OL at the ending. It has all those hydroxyl groups on it. So it would be in that alcohol category, because it's of functional groups. This is the backbone that forms these triacyl molecules. And the fatty acid is the one right below in this box. So here you can see right here a carbon, double bonded oxygen, and an OH. So this is a carboxylic acid functional group. And then if you look at the rest of it, you say, wait a minute, there's a long chain of hydrocarbons. And that's where a lot of this energy storage is. So down below, this triacylglycerol is a combination of those 2 things above. So here is the glycerol, and here's the ester linkage. And we've lost on each of the molecules, but between each of these sections, we've lost a water. So you can see this fat here that is formed, and this lipid, which in this case, because everything in here is single bonded, we have a maximum number of hydrogens, it's going to be a saturated fat. So that's the definition of saturated, is that every one of these carbon chains on this part of the molecule have the maximum number of hydrogens possible because there are no double bonds. So here's an example of saturated versus unsaturated fatty acids. The one above is saturated, because what you see on that piece. This is a fatty acid that will join to that glycerol backbone, but there are no double bonds. And so saturated fatty acids are able to pack more tightly together, which is why they can exist as solids at room temperature. Not particularly good for us if we have too many in our diets. Here's another fatty, a common unsaturated fatty acid, which is oleic acid, this one right here. Notice the double bond right here smack in the middle, and that means that we have fewer hydrogens, making this unsaturated, in part because, and also making this be much more of a fluid fat, so more like an oil, and that's because the double bond actually creates a kind of a kink in this molecule, which doesn't allow it to pack tightly together. So most unsaturated fats are liquids at room temperature. Here you can see that the kink that I was just talking about here. So now we can look at it and use the term cis and trans that we talked about earlier. So cis acids, ones that have this formation right here that you see in the middle, where the hydrogens attached to those carbons that have a double bond are on the same side, those are the liquid types. This is the cis type that we kind of prefer in our diets. Where the trans, because of the symmetry of this molecule, these trans fatty acids are ones that are very often made actually artificially by forcing in these hydrogens in these particular locations, and those things aren't good for us. So foods with trans fats aren't as healthy. So these also would be much more solid at room temperature. This type of fatty acid is one that we like. These are the omegas. So omega fatty acids have these really interesting structures. Notice that there are double bonds. Here's a double bond, here's a double bond, here's a double bond in the way this is shown in this molecule. So there we have omega-3s, we have omega-6s, and those double bonds, which are more double bonds than even the other ones we just looked at, are much healthier for us. And we find these very often in things like fish. Let's see, makes up fish fats. Waxes are useful in nature, particularly because they have these really long fatty acid chains. And when it says here esterified, it just means that those are ester bonds, ester linkages to longer chain alcohols. And you don't need to know the actual chemistry of this, but basically waxes are things like what you see here. They can coat the outside of leaves, preventing dehydration, preventing things from sticking. So very helpful. Phospholipids we'll go into detail on. So these have, they're a group that have both a phosphate, remember that it's also a functional group, has this glycerol, again, just the one we saw before that's related more to alcohols. And then it has only two of these fatty acid chains, one of which has a kink in it. So you can see the unsaturated and the saturated. We'll talk more about these in some detail. They're critical in our bodies, so really, really important. Here we can see a little bit more on that, getting a little bit of a better picture. On the left is just a structural view of it. On the right you can see that space-filling model. And then on the very far right, what you see is sort of the typical way we draw them. So you'll see me draw them like that. They make up the membranes around cells. So this is why they're so important. And because membranes are double layered, they actually form what we call a phospholipid bilayer. The hydrophobic parts of that are going to orient themselves toward the inside to get away from water, because out on the exterior here around a cell and on the inside that would be like where the cytoplasm is, it's very watery. And so these are hydrophobic, so they try to get away from water, so they turn inward. The term for a molecule that has this dual nature, where part of it is hydrophilic, the phosphate part, but some of it is hydrophobic, is called amphipathic. So like if you think ambidextrous or an amphibian or something like that, it's that idea of dual nature. So this has a dual nature. It has a hydrophobic and a hydrophilic head to it, hydrophobic tails. You can see that. Steroids have ring structures, and you can see what they look like here. So steroids are very important. Here's cholesterol, which is one that we need. We need cholesterol. There's good kinds of cholesterol and bad. And then there's the one below it, which is a hormone that's a stress hormone called cortisol. So these are both hormones that we see, and they're under that category of lipids. They too are going to be hydrophobic, which means they don't like water. And the thing about cholesterol in particular is interesting because we don't really know a lot about it. We just hear about cholesterol a lot. Is that it's made in the liver, but it's an important steroid because it is this precursor. In other words, it's the sort of that forms the basis for other hormones to be created. So things such as testosterone, which you're familiar with, I think, and estradiol, which is, you might think of it as estrogen. It also is important for the production of vitamin D and bile salts, which are things which you'd find in your gallbladder, which help to actually break down and digest fats. And that's it.